Hi, hello everyone. This time, I'd like to talk with you about the process of protein synthesis, right? Okay, I'll start with the ribosome, which is called S80S, okay? And the first step of initiation of protein synthesis, this ribosome need to be dissociated into two subunits. Okay? In this process, we need a small protein called eukaryotic initiation factors. And I have a legend on the right bottom part here. The blue will tell you those are the ribosome or ribosomal subunits. And this green circle with a number that indicates eukaryotic initiation factors. Okay. Here we have initiation factor 1 and 3 and 6. Okay. Now we are ready to initiate the translation. And these initiation factors, 1, 3, and 6, will cause the dissociation of 80S into smaller subunits. One is a 60S, and the other one is 40S. This is done by, for them binding to the subunits. Okay. Initiation factor 1 and 3 will bind to 40S subunits, whereas initiation factor 6 will attach to 60S subunit. Now, these ribosomes are dissociated, and now they can be activated, attached to messenger RNA, allowing tRNA to come as well. Okay. So this is the beginning, right? And I'll put 60S with initiation factor 6 away here. And I'm looking at this 40S subunit with initiation factor 1 and 3. In the cell cytoplasm, at the same time, there are amino acids. So this yellow circle with AA means free amino acid. And in this case, this is dedicated to methionine. Also, in the cytosol, you can find the uncharged free tRNAs. So there are 20 different types of tRNAs existing, responsible for corresponding amino acid. So there is a tRNA dedicated for lysine. There's a tRNA dedicated for methionine, and so on. Okay? So you need 20 types of tRNAs. First thing need to be happen here is activation of tRNA. And let's go through what happens here. And in this activation, we need another initiation factor called initiation factor 2. This initiation factor 2 include GTP, guanine triphosphate. Okay? It has energy. Okay? This will cause the formation of something called ternary complex made of 3 components, which are initiation factor 2 with the GTP, and amino acid, especially in this case will be methionine, and the tRNA responsible for the methionine. Okay. So this is a ternary complex. tRNA with methionine called methionine tRNA with initiation factor 2 with the GTP. The next thing happening is related to messenger RNA. So I just draw one messenger RNA here. On your left side, there will be 5' methyl cap. On the right side, this will be 3' end, which is made of adenine. So we call it poly A. Okay? So 3' poly A. And then this messenger RNA need to be activated. And here, there are several components involved. First, PAB. PAB is poly A binding proteins. So those are the proteins will recognize the terminal site of messenger RNA, 3' end, and attach to this poly A. On the other side, we have additional initiation factors, including 4A, 4B, and 4E, 4G, and then another one is called 4E binding protein 1. And we need a phosphorus. And next thing happening is phosphorylation of 4E, 4E binding protein 1, and 4B. These three components need to be phosphorylated to be activated. Right? So now this 4E and 4E binding protein 1, in the previous slide, they were bound each other and they are inactive. They cannot do anything and they are bound each other. And this phosphorylation will dissociate them. So 4E is now, it can form another structure called 4F together with 4A and 4G. At the same time, 4B is phosphorylated as well, and they are activated too. Next thing happening is to the messenger RNA, the poly A binding protein are attached to poly A site at 3' end. At the same time, initiation factor 4E with phosphorus, 4A and 4G, they are bound together, forming initiation factor 4F. Okay? So 4F is bigger compound composed of 4E, 4G, and 4A. And there is a phosphorylated 4B as well. Okay. Next thing happening is activation of messenger RNA. Okay. If I go back, okay, this is the messenger RNA with 5' methyl cap and 3' poly A end. 4E, this is the initiation factor, which will find the 5' methyl cap and bind. And 4G will look for poly A binding protein. In the meantime, 4A is an enzyme helicase, which will make a space for the translation. And this is the outcome. So 4E is bound to 5' methyl cap, and this 4E is still phosphorylated. And the 4G is bound to poly A binding proteins at the end of 3' end. So this messenger RNA forms a kind of loop. Okay? As we talked in the class, typical length of this one messenger RNA is made of about 300 to 1,000 nucleotides. Okay? Consider that 3 nucleotides make a 1 codon, so you can kind of estimate how many amino acids can be added to this from this translation process. Okay, now going back to this ternary complex. This ternary complex will be bound to 40S subunit with initiation factor 1 and 3. And that will cause the formation of 43S pre-initiation complex. Okay, and this is what happened. So here we have methionine tRNA with the initiation factor 2 with the GTP bound to 40S with the initiation factor 1 and 3, becoming, this whole thing is becoming 43S. Now we have 43S, and also we have activated messenger RNA. Oh, it seems like we are ready to do translation. Okay. Now this 43S comes to messenger RNA complex, and all together, this will form 48S initiation complex. So we call it 48S initiation complex. Please note that in the cytosol, there are many free amino acids available, and they are originated either from diets or from the protein degradation from other tissues or from the within a cell, right? But these are the substrate for making polypeptide. All right, the next thing happening is this methionine tRNA with 43S forming 48S complex. This will start the scanning. Will move around the messenger RNA until it finds a start codon. Start codon is with a nucleotide AUG. So AUG is called start codon, and the scanning will take place, spending energy, and this 48S complex will stop. Will find the start codon and stay there. Okay. And so this is what happened. And now this ribosome found the start codon. Seems like it will start the translation. To do that, we need the 60S coming back to for the association rejoining of the subunits. Okay? So this 60S will come. And at the same time, the new initiation factor called 5, this is initiation factor 5 with the GTP, they'll cause the dissociation of all initiation factors, 1, 3, 6, and 2, allowing 60S to come in and join together. Okay? So that's what's happening, this subunit joining and recycling of initiation factors. All right, so those initiation factors are gone for other translation, other initiation of translations, and then the 60S came and combined with the another subunit, making 80S initiation complex. Now you see initiation factor 5 with the GTP. This 80S will provide a space for the new amino acid tRNA will come. Now the initiation factor 5 will be detached and the GTP is used up and it will remain as a GDP. At the same time, initiation factor 2 used up the GTP energy. Now it has a GDP and then need to be converted to GTP for the next initiation of translation. And this process is coordinated by initiation factor 2B. Right? So this is another important process here. Okay, with this, now we are ready for elongation, the making of polypeptides. I will try to draw this ADS again in more details. Okay. You look at the ADS initiation complex, it can read 3 codons at one time. The location where the first codon is read is called the E site. That's a short name of exit site. The second one reading actually the start codon is called P site. That is short name of peptidyl site. And the third area for reading the third codon is called A site. And that's a short name of amino acid site. So there are 3 sites in ADS for the tRNA to come and stay. Okay, now if you look at the left parts of the slide, I put the a free tRNA. Those are uncharged tRNA, which means tRNA without amino acids. Okay? They need to be activated by binding to designated amino acids. And here I'm introducing new eukaryotic elongation factors called eIFs. So elongation factor 1A with a GTP and elongation factor 2. So they have all the role here. Okay, as I just mentioned, this tRNA will be activated. So it's called amino acid activation. So uncharged tRNA will be bound to corresponding amino acid. So if this is a glycine, as an example, this whole structure will be called glycinyl tRNA. If this is a serine, okay, and serinyl tRNA, and so on. Okay, this process consumes energy. Right? You can see that from the ternary structure, this ribosome already have methionine tRNA, and the tRNA will be located in the peptidyl site in the middle of this ribosome. Okay? That's the beginning of the elongation. Now this elongation factor 1A with the GTP will carry next amino acid tRNA following the codon. So whatever the codon says, if this is coded for lysine, then this should be lysinyl tRNA carried by elongation factor 1A with GTP. So this lysinyl tRNA will sit on the A site, which is an amino acid site. Okay? So that's what happened. Now there are two amino acid tRNAs. The first one on P side is a methionine tRNA. The second one newly came. I made a case. This could be lysinyl tRNA, or it can be anything depending on the codon here. Next thing happened is methionine on the P side, which is from the methionine tRNA, will be attached to the amino acid, with tRNA just came to A site. So there's formation of dipeptide, methionine with a new amino acid. So this tRNA, which had methionine at the beginning in this P site, is free, okay? It's uncharged. And then now new elongation factor 2, which is with GTP, will move the ribosome from one codon by one codon. So from the 5' towarding 3'. So this whole structure will move to the next codon, okay, like that. Previous one, now move to the next site, like that. Okay? This process will consume GTP. So this GTP attached to the elongation factor 2 now became GDP. And earlier, when elongation factor 1A brought a amino acid tRNA, it used GTP. Now it has a GDP. So there are energy spending here. So by moving ribosome to a next codon, that'll cause the changes in location of tRNAs. The first tRNA, which had methionine at the beginning in the P site, now it's moved to the E site, X site. The second amino acid tRNA, which was just came in to A site with the help of elongation factor 1A. So now it is in the P site. It carries the methionine from the previous tRNA too, forming the dipeptide. And then this tRNA in the exit site, E site, will leave the ribosome. Okay. So it looks like this. Now, this ribosome has a tRNA in the P site with 2 amino acids. Now it's ready to bring the next amino acid tRNA following the codon. So this could be any amino acid based on what codon you have here. But the start codon, AUG, which was in the P site, is now moved to the E site, okay? Because ribosome moved from the 5' to 3' by one codon. Right? I'm repeating the previous process. For doing that, elongation factor 2 with the GDP need to be charged again with the GTP. So it's now ready. Also, elongation factor 1A with the GDP need to be charged back to GTP. Now you can bring next amino acid tRNA to the A site. Okay. And this GTP will be changed to GDP and leave the ribosome. And now I'm repeating here. This is a dipeptide with methionine and the next amino acid attached to tRNA on the P site. Those dipeptide will be attached to the new amino acid with tRNA on A site. So forming a tripeptide. Okay, so now like that. Okay. But at the same time, elongation factor 2 with GTP will move the ribosome again to the next codon. Okay, so we'll move like that. Okay, again, like that. So what's going to happen next? This tRNA, which transferred the dipeptide to the new amino acid tRNA, is free now, and it is on the E site, exit site. So this tRNA will leave. Okay. All right. So same thing happened, but the difference is this tRNA in the P site has a longer peptide. Now it's tripeptide. This will repeat it again and again and again until the ribosome moves to the stop codon. Okay? This one is one of the stop codon UGA. Okay? So it will be repeated. As this elongation happens, this peptide will become longer and longer and longer based on the codon. Okay? All right, it's continuing. So polypeptide is growing. And this is the reality. What really happens in one messenger RNA? If this is a messenger RNA with 1,000 nucleotides, okay, there probably you can find 8 to 9 ribosomes, 80S ribosomes. Okay? And it will start from the start codon and then the translation, elongation until stop codon. Okay? So by doing that, there are multiple proteins made at the same time. Going back. Now this 80S ribosome found stop codon, UGA. Okay. This will stop the elongation at A site because there's no tRNA bringing any amino acid into UGA because UGA doesn't code for any amino acids. Okay. So this will cause the empty space here. And then now I have a release vector, something called release vector. In other words, it's called terminating vector, termination vector. So ERF 1 and 3, ERF 1, ERF 3 with a GTP. So you can see that this process also needs energy, will spend energy. So release vector 1 will recognize this empty site, empty A site because of UGA, and will sit on that site because there's no tRNA coming. And then there's a release factor 3 with the GTP will bound to this release factor 1 to stabilize. And then release factor 1 will cause the hydrolysis of the bond between the amino acid and tRNA on the P site. Okay. So this polypeptide is ready to depart the tRNA or ribosome. Okay. It will go to any destination that is determined, okay, which we will learn later. Okay. So now there's a polypeptides need to be modified to become a protein. Now this releasing vector will depart ADS. tRNA will leave also. So this ADS unit, ADS ribosome is empty. And this will be reused when the initiation factor 1, 3, 6 comes. Okay? And that will cause the dissociation of this ADS into 60S and 40S. So that can be, they can initiate another translation. All right, so this is all. And I hope this slide helped you understanding the process of protein synthesis better. Okay? So thank you for your attention.